cosmic storm. Floating on whimsical wavelengths is the norm. Dancing through the stars, chasing spectrums of light. Riding the waves that are just out of sight.
Welcome back. I love doing this podcast because I learn new things every single time. The fascinating universe we live in, and today is going to be into something pretty obscure. Before you groan and suggest every episode is about something obscure, well, maybe you have a point, but no matter. The angle of understanding the obscure can have very clear impacts on how we understand the world. Okay, to get to it, this is another trip to entomology, looking at insects. In this case, a specific group is called the twisted wing parasites in the common parlance of people, if that ever comes up, or its name, group and order name, Strepsiptera. It's a relatively small group by insect standards, with around only 650 species described. And of those, 43 we actually only know from the fossil record, because they got trapped in amber specifically. Now, for a bit of a detour, but an apt one, Aliens the movie. The aliens were designed off of parasitic wasps, where they have the young burst out of the insect that they've been parasitized. But these wasps have some visceral similarities to Strepsiptera, or twisted wing parasites. See, it's a parasitoid, an organism that spends a significant portion of its life attached to or inside a single host, which eventually either kills or sterilizes the host. I think most of us find the movie Alien terrifying, or at the very least disturbing. But for millions of insects on Earth, that movie could be a documentary. In my background for the episode, Strepsiptera was also referred to as an endoparasite. More broadly, that means that they live inside a host's body, like their intestinal tract and tissues or blood, deriving nutrients while causing harm, such as malnutrition, diarrhea, or severe infection. The other term above, parasitoid, is a subset of something more extreme. Just to clear it up now, these little guys, Strepsiptera, do not infect humans. But there are others that do, like tapeworms, the malaria parasite, and a whole bunch more we don't need to talk about today. Strepsiptera only infects insects, generally living in their abdominal cavity. From evolution to how they accomplish this, it is just incredibly strange from a mammalian point of view. And more importantly, these little insects have big shoes to fill scientifically. Understanding their evolution has been complicated, from their lineage to their DNA. Understanding them used to have a moniker, the Strepsiptera Problem. What is the closest group that shares a closest common ancestor? What was it? Is it? It's a problem we will discover the answer to here on the episode. But it has been a very contentious problem within academia. They are also a help to us humans, as they parasitize many insects that we call pests, and are super important for the food web. These little aliens might be the reason your local farmer doesn't have to spray as much pesticide on our food. There is one more technical term to mention, which is behavioral hijacking, which is, at least to me, brings up the idea of zombification, basically getting the host to do something they wouldn't do otherwise. Before checking your own skin, breathe. Humans do not have parasitoids. We're too big, too complex, and our immune systems are too loud. Insect immune systems, on the other hand, are much simpler. Of course, I could not attempt this topic much further than this intro without a guest. Okay, please welcome here to Whimsical Wavelengths, Dr. Rebecca Jean Millena, who is currently a postdoctoral fellow in Biology Department of the University of Rochester with the Trop Bio Lab, and obtained her PhD from the Comparative Biology PhD program at the American Museum of Natural History's Richard Gilder Graduate School with the Ware Lab. Yay! The first question is always, often at least here on Whimsical Wavelengths, is how did you how did you get into your specific STEM field, science field? Because it's quite varied, and I think listeners should realize that not everyone is just born a scientist. Some of us discover it early, some of us discover it late. and discover there's distinctive fields only after getting to university. Which were you?
Sure. So I was one of the lucky ones who was kind of born. My parents like to joke that I was born without a sense of um, survival, a good sense of survival, because even when I was little, I would just pick things up that were crawling on the ground because I thought they were exciting. So I have been obsessed with bugs since I was quite small. And it worked out really well that my parents, instead of getting really freaked out about it and kind of banning me from picking stuff up, they instead bought me books so that I could be informed about it and pick stuff up safely. So that encouraged my love throughout the years, and I got lucky in that entomology is a profession that I could aspire to. So I kind of was the token bug kid all throughout grade school years, and it just compounded.
Well, it sounds like your parents did a pretty good job. I mean, as a parent nowadays, and myself, I got two
Oh,
little monsters. It's always like trying to give them the tools so they don't kill themselves.
yeah. I think that's precisely what happened, so I'm very grateful. There was a time though where my first pet kind of officially was a tarantula, a gift from a family friend, and it did get loose, and then I was banned from
Ooh.
having tarantulas in the house, so there was a balance.
But you had lots of bugs in the house at one point, I would imagine. The
Oh,
ban was
yes.
eventually lifted.
Yeah. Well, it kind of moved to the backyard, so
Oh.
instead of having tarantulas, I ended up staying smaller and staying local. So there were a lot of box elder bugs from a tree in our neighbor's yard that would make their way into our yard. So I kind of grew a colony of those in a recycling bin for a long time. My sister and I called them tappers, and then the same with ladybugs. I would just kind of keep colonies of things.
Okay, so I'm going to come back to that colony of
Yeah.
bugs in a moment because I think I have a question with that colony of bugs and our current subject today. So, in the intro, I tried to set the stage, if you will, but I want to hear it in your own words. Who is the subject of your PhD research and the episode today?
Yes. So my favorite bug, my favorite insect, and the thing I have essentially dedicated my life, hopefully, to studying is Strepsiptera. The common name of this group is the twisted wing parasites. So Strepsiptera means twisted wing, and that's the Latin name for the order. And they comprise something like 650 or so species of really bizarre little twisted wing parasites. So they
Is 650 a
parasitize.
large number or a small number?
It's quite small. So, in relative to other insect groups, if you think of things like beetles, which incidentally, maybe this is a spoiler, you can cut this out.
No, 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 we're going to keep going. We can meander
Okay.
every which way. This is the
Oh,
messy part of
bless.
science and discussions.
Excellent. So, in regards to size, when you think of Strepsipteran's closest relatives, which are beetles, the consensus is that they're derived from or most closely related to beetles. Beetles are an order of something like almost 400,000 species. So when you think of Strepsiptera having 650, you can see why I might think it's a tiny order.
Right. Okay,
It's very
so
fun.
what makes them unique or special though? Like you've dedicated your life to them. There must be something about them.
Many, many things. Strepsiptera is fascinating. They
You
break
almost
a
asked
bunch
me like,
of.
how much time do we have?
Yeah, I could probably talk about them for many hours. There are just so many facets of them that I can't believe are real. I don't know. They're stranger than fiction. The fiction stranger than fact in this case. They have such a distinct look. They're very iconic, at least the males, because they have these like raspberry looking eyes. Like insects have compound eyes, but with Strepsiptera they have even more bold, like they look like raspberries. They're described as berry eyes a lot of the time. And then the thing they have that kind of no other insect does are the twisted wings that they're named for. Their forewings are kind of these club-like structures in the males, and the closest thing that they look like or the closest thing to them in other insects, are fly hindwings. So for a very long time, people thought they were related to flies, and they have fan-like, Strepsipterans have fan-like hindwings, which is also very unique in that the whole group has that. The biggest thing that they're known for, and that is kind of sensational, is the sexual dimorphism. So the males and females look very different. That's what dimorphism means. The males are that iconic kind of insect that I just described, but the females are basically worms, and they live and die within their host. So both of the male and female Strepsipteran, they both develop within another insect, which is what makes them endoparasites or parasites of other insects. But the male will eventually be able to fly out of its host, whereas the female stays there forever.
So many questions. So many questions.
Yes, of course.
Let's start with when the male, when it flies out, let's skip everything, all the other questions, because you just
Sure.
focus on that one little bit. When it flies out, does it also kill the host, or does it somehow survive?
So the host can stay alive for quite a while afterward. There's actually ongoing research with this happening in multiple facets, but something that's very fun is that my current lab has a couple different projects running on survival. And this is possible, just to plug how incredible my current lab is. We have lab stock of Strepsipterans, which is something that is very difficult to do, because you have to be able to rear their hosts as well as the parasites. So you need to know two different life cycles and how to keep them alive. So I'm stoked every day at this system. But yeah, we find that the hosts do stay alive for a while after the males emerge. And anecdotally, I've also seen the same thing with a couple other species. When the male has left, the host still lives for what sometimes I think is the natural life cycle. And there's also a chance that they might die on chance prematurely because of infection that might come in through the wound that the parasite has left.
So when I was doing my background research for this, the movie Aliens came up.
Yes.
I realize it's not the what is the word, the muse or the inspiration behind them. Those were the jeweled wasps, I think. But nevertheless, this is like, some have termed it the alien model.
Yes.
So there's lots of these, and they don't infect humans, right?
They do not infect humans. I know, I love thinking about that. I have given actually a couple talks about the parallels between Xenomorphs and Strepsipterans. So last year in 2025 at the Museum of the Moving Image in Brooklyn, New York, they held an event called Creature Feature: The Science of Monster Movies and Community Filmmaking. And the screening that they showed that afternoon was Alien. And I got to give a half-hour-ish long talk about how xenomorphs can be seen in real life, like in nature, and how those mechanisms exist already. We don't have to look into space to find them, they're already here. And I took the opportunity to kind of hijack it and use it as a way to brag about Strepsiptera. So it's perfectly on the nose. They are really good explanations for each other.
Okay, so just to kind of go down the life cycle a little bit more.
Yeah.
Let's pretend we already have a male and female. The male bursts out like a xenomorph jumping out of a human's chest, which is visceral and scary. It has these raspberry cute eyes and these twisted wings, and it flies around, and what does it do? Just kind of go, hey, there's another ant. I'm going to go pester it and hope there's a female inside of it. Like, this isn't like, it's not a cricket. It's not making noise. What's going on?
Yeah, that's a great question, and something that is very fun to answer, because it turns out they have really good sense of smells, basically. Senses of smell. They have a ton of sensory organs, kind of concentrated in their antennae, but also on their legs and a lot of sensory hairs, but they can smell the female, essentially, with pheromones. So she puts out a really strong chemical signal, and the male can pick up on it.
Wait, so let's go back to my idea or my example of an ant. Because they can infect ants, right?
Yes, but actually, it's fun you bring that up because only the male infects ants, and then the female of the same species that infects ants. The female is found in crickets, katydids, and other orthopteroids.
Okay, so the females do not infect insects that live in a hive.
So there are many other species that are best known actually for infecting insects that live in hives, but they're in a different family.
Right, I meant specifically these ones. Because I was curious, because if your female's producing all these pheromones that smell to get the male, and yet that ant then walks back into the nest, they should smell like the female. That's where I was going.
Oh, I see. So the male has left the ant.
No, I know, but you just answered the fact that the females don't infect the ants, although
Oh,
the males
yes.
can.
Yes.
We'll get to that, how
There,
they
it's.
choose their hosts, I guess, in a moment.
We don't even know.
Okay, so
Yeah.
somehow the female produces pheromones outside of the insect that she's living inside.
Yes, okay. So something I didn't mention, thank you for asking this for clarification. Something I did not mention is that the fun thing about Strepsiptera is that they protrude a little bit from their hosts
Gross.
upon maturity. Yeah. So I'm glad you asked that because I kind of take it for granted
So it's
that.
just like a tumor that sits on their belly that
Yep.
just kind of. Oh,
Yep. Yeah, that's almost exactly what it looks like. There's just a lump in between the host insect's segments, basically. And for the female, which stays there, that's her head. Her head is just kind of a little bit outside of the host, and she can emit pheromones that way.
wow.
Yep.
Does she also breathe that way, or does she breathe from inside the insect?
For a large part of the development, the female is getting oxygen from the circulatory system of its host. So I believe that actually does not change. I don't think they have spiracles on their cephalothorax, but I could be wrong.
Neat.
Yeah.
So they have a whole separate way of breathing than most other insects because they're living inside an insect.
Yeah.
But yet the males still must be able to breathe like other insects do.
Yes, upon maturity. So while he's within the host insect, it's the same kind of system as the females. But as soon as he pupates and emerges, he has to be able to breathe on his own.
Okay, so he finds the insect, or he finds the female, trapped
Yes.
it in by the smell, and the other insect just goes, "Hey, yeah, let me present my abdomen to you." And the female bursts out. Like, how does this all work?
So, yeah. So the female is, at that point, as emerged as she can be. Her head is probably the furthest out that it ever will be in her life because she's calling for a male. She's sending out pheromones. The host insect, depending on what it is, may or may not have been influenced to be somewhere or stay alive for certain timing to optimize this encounter. So if it was a hive insect initially, with the parasite developing within it, it is now not acting like a hive insect. It'll act like a
Zombie?
perhaps. Yeah, a little bit. Yeah. They won't necessarily participate in the same kind of nest activities, which is very fun. And they will still, though, video anecdotal evidence given, the host will still try to kick off the male sometimes. Like, it doesn't necessarily like something else tiny buzzing around its abdomen.
Right.
So there is still risk that the male will get fended off from the female by the host, but the males are actually very zippy. So they're very quick and agile, and I like watching videos of them trying to get at females because it's really interesting.
Okay, so they manage to find a female. They impart their genetic material through the head that is extruded through the abdomen.
Yes.
And then the male goes off and dies?
Yes, precisely. The a fun thing that I would like to mention about the imparting of genetic material that can happen through something called traumatic insemination, where the male just sticks his genitalia anywhere in the female.
Interesting.
Yes.
And how does the genetic material get to where it needs to go?
It goes through her hemocoel, her blood, essentially, and then it just makes its way to the eggs.
Is that a common occurrence in the animal kingdom? Or is there anyone else that does it?
Yes, in fact, yeah, bed bugs are well known for it.
Oh, gross. Another reason to
I know.
Very nasty. Oh my goodness.
And now you know.
Now I know.
Mhm.
Okay, so the male goes off and either mates with another one or doesn't and dies. And
Yes.
now surely the female ends up killing their host because now they have to rear a whole bunch of eggs too, right?
The host is still alive.
At this point. But then what happens to, which I assume because it's an insect, it lays eggs.
So they
hatch.
do something called ovoviviparity, where the eggs essentially hatch inside the female, and she becomes a bag of larvae.
So she is the host for her parasitic young.
Yes. So that is called also technically matrophagy, or eating mother, where the mother is entirely consumed, at least in this case resource-wise, by her larvae. And she isn't actively laying, actually. They're just crawling out of her head.
Wow. And she
Yeah.
survives this?
Yeah. She keeps pumping out approximately hundreds of thousands, like 750,000 is one of the recorded numbers in literature. That's how
would you
many
even
babies she
measure
has.
that?
That's a great question.
You don't know the answer to that?
No, I've tried. I have collected a gravid or pregnant female before, and I did sit for a very long time just watching the larvae crawl out of her because it was fascinating. That was the first time that I'd been able to witness it, and over the course of even just a couple hours, I could not keep count. There were hundreds.
Okay. So going back to your bug collection, did you ever find
Yes.
one of these in your bug collection?
Yes.
So as a kid growing up, you saw this?
Oh, no, I did not know. So that's a great question because it leads back into kind of what brought me on this journey. I'd always been obsessed with bugs, but the way I was introduced to parasites specifically was at UC Davis, my alma mater for undergrad. I was impatient to start taking entomology classes, so as a freshman, even though I couldn't take my upper div entomology courses yet, I enrolled in a general ed entomology course taught by the grad students, where every week was a different topic. And Jessica Gilling, who at the time was a TA, she specializes in parasitoid wasps. And watching her lecture was the most excited I had felt in a very long time about insects. I was always excited about them, but that one kind of was like a lightning strike about how cool they were. So I ended up doing my final project for that class on parasitoid wasps, and then every other entomology course I took after that that had a field component or a final project component I did on parasites.
Okay. But you didn't find them in your insect menagerie, if you will. Okay, well, I think we're going to learn a lot more about Strepsiptera. We're going to learn a lot more, but I think it's time we now introduce the paper. And in full, just being open, in episodes that weave together the past, the present, the future, I struggle to keep the episode length down, especially when I know so little about a topic, and I'm already asking way more questions than I had planned. And I had a sense that was going to be the case, but it's also a terrible segue to say that the paper's title, Strepsiptera's Systematics, Past, Present, and Future. I guess let's start with the past. What makes this group, in the words of the paper, among the most obscure and enigmatic orders? I mean, we've already talked about why it's so cool and special, but why is it so obscure?
Yeah. So Strepsiptera as an order wasn't recognized as such until 1813 by this entomologist William Kirby. Before then, no one really knew what it was. It was grouped with Hymenoptera, which is the order of bees, wasps, and ants. The first time it was ever seen in, I think, 1700s, late 1700s. And after it was seen, people didn't really know where to place it because before DNA data was available or even recognized as a thing that existed, we all had to depend on what things looked like in order to group and name them. So Strepsiptera, as you can imagine, look pretty bizarre, not to mention the fact that the males and females look the way they are. So you can kind of imagine also that it was very difficult to figure out where they belonged in the tree of life that was forming at the time.
But was it an obscure thing because they didn't know where to put it, or because they were so hard to find?
Both. Definitely both. So those are the two big kind of famous obstacles to studying Strepsiptera in general. It's hard to describe something that you can't find much of.
Right.
And yeah, they're tiny too.
Was there a difference in age about when the males were discovered versus the females?
That's a great question and something I don't have off the top of my head, but I'm pretty sure that, um, yeah, I don't actually know that in specific. That's a great question. I mean, it is known for the myrmecolacids, the ones where the males infect ants and the females infect katydids, crickets, mantises. Those ones, there was a 94-year gap in between the description of the species versus the reunion of the male and female, at least in our taxonomy. But regarding not the whole order, but the separation of the sexes as an order, I will look into that because now I also want to know.
Wow. If you give it to me before I get around to recording the intro, I'll add
The
it there.
file. Okay, that would be awesome. Yeah, I'll double check because that's a really great question. I've only ever looked into the history of the order as a whole in taxonomy.
Just to go a little further, I mean, in my background, it seems to be that some species are only known from the males or only known from the females, so we still haven't merged some species, male and female. So
Yes, a great many.
it's crazy to think about it. Yeah, we found a lot of the males, but we have never found the females. And we found a few females over here because we dissected this creature and found them, but we have never seen the males. You're like, wow. You know, it just goes to show that there's so much more we can learn and understand that we just don't know about the natural world around us. I mean, we're still finding new species and stuff too.
Oh, 100%. Every day.
Every day, every single day.
Every day.
But I think we're also kind of dancing around what the next question is, because I read this in multiple papers in the background here, especially the older ones, not so much the newer ones. The past understanding. It was also given the Strepsiptera Problem, and I'm sorry if I automatically pronounce it wrong.
Very all right. Yeah, I mentioned earlier that Strepsiptera was so infamous and it was difficult to tell like what it was grouped with. And that was given its own name. As you mentioned, the Strepsiptera Problem is the title given to this dilemma of not knowing what they were. The problem itself was the question, what is the sister group to this species or to these animals, these weird little parasites? What did they arise from evolutionarily? And it was a problem because there were just no good consensus.
Did anyone just throw their hands up and go, aliens?
Honestly, yes.
Oh, really?
Yeah, there were some. It's very funny looking at very old, or not very old, but the past literature. Um, I guess it is very old because it's been like 200 years since some of them. Or not quite 200,
Yeah.
more than 150 years for some of them.
Well, you got to remember that my modern day science, geology, geophysics, really only got going in the 60s. So,
Oh, that's fascinating to me because I would think that it's been around forever, you know? So like,
yeah, but when you don't understand, can't map the seafloor to see plate tectonics, and
mm,
you can't measure the age of the Earth until you understand atomic decay.
that's a really good point. This is cool because yeah, it's the same kind of thing where I don't know as much about the physical sciences. So hearing about this and those logistics are really interesting.
Yeah, so in this particular case, though, we're going back to the topic before we get completely derailed into a
Yeah,
history
understandable.
lessons about geology and geophysics. Um, so people didn't have a good idea of what these things were or how to put them in the tree of life. And I gather this all comes down to the theory of evolution, right?
Yes.
And so where people like, okay, the theory of evolution is broken, or did they just go, we're missing a key piece? Like,
It was more the latter.
okay,
It
good,
was,
good.
yeah, I know. It would be funny if we could blame Strepsiptera for kind of a breaking of theory. But no, it was essentially that it seemed like there was a missing link because there were kind of common characteristics that they seemed to share with some groups, but not all of them. And for some people, some sharing of traits was enough evidence, but not for a lot of other people, and vice versa depending on the group. So it was a very contentious debate for quite a while.
So, what were the main candidates here? You've already mentioned beetles. I think you mentioned wasps and ants.
Yeah, yeah, the first group that they were placed in were wasps, and my thought and my understanding was that because they were seen to be parasites, there are a lot of parasitic wasps, and because they fly, etc. Um, and then the other thing was flies, because they look like flies and they have that little club-like structure, the twisted wing, which is the haltere in flies. That's what it's called. So, given all of those were kind of the three main groups that people figured they should be in.
Is there anything at all analogous to having that dilemma that you're aware of in science in the
There
last
are,
200 years? Where you're stuck going, is it one of these three really large? I have the creature right here, but I don't know. Like, that
yeah.
seems very unusual in, let's say, even by the 1920s, right? Like,
Well, it actually happens a lot more often than you think. Just because I'm immersed in the field of taxonomy, the field of taxonomy is very dramatic.
oh my
There
goodness.
are, yeah, there's a lot of this, especially in paleontology, but in essentially every group in the animal
Well,
kingdom.
paleontology I understand because we don't.
Yeah,
We just,
they're still at appearance because,
yeah,
you can't really do anything
but the
else.
live ones we can observe them. We can observe them move and how they fly or not fly, and I would just expect that the stuff living today would be no.
No.
It's
Yeah. So
okay.
it's a very human judgment field. It's surprising because it's one of those things where you think, oh, we have such overwhelming evidence. We have so much technology. We can record everything that these animals do. But the thing to remember is that taxonomists, the people who are determining what is related to what and what is a species, we are all human. And humans have opinions. So it can get surprisingly heated.
So what you're saying is it's not completely qualitative.
Yes,
Quantitative, sorry. It's a little qualitative and not completely quantitative.
sometimes. It is very rigorous, I have to say. It's not
Sure.
just like I'm discounting, yeah, all of taxonomy, but the thing about it is that you have to be very, very specialized and detail oriented, or you don't have to be specialized, but it ends up being the case that taxonomists become very specialized because in order to describe something, you have to know it inside out. Literally, a lot of the time, morphology inside and out. And taxonomists, to describe a species, have to choose the characters that they judge with their best educated judgment to be the things that are informative about what sets this group apart from everything else. So that involves staring at an animal or a plant or a fungus for a very long time and determining the tiny little differences between it and something that looks really similar to it, except for this one little character.
Okay. Well, we still use this technique, obviously, today, as I just learned. But we do have some modern stuff as well, right? Like, now we're kind of diving. So we just talked about the past and how it's muddied the past on what it
Yeah.
was, what is it related to, hard to find. Not many people talked about it just because it's not causing us problems. And we'll get around to why it's a good thing for us overall, probably closer to the end. But at some point, science moves. We get new tools to conduct science and collect data. And we started to be able to sequence genomes. So I assume this is, I guess, two parts. One, how does it change the dynamics? But first, how does that change taxonomy? How does that change that process? Because now you have something that is very data-driven, that has no subject matter. Like it's this and only this. How did that change this whole classification scheme?
Yes, wildly. That's also a great question. So, DNA and genomes, but starting with just like even singular genes
Yeah.
being sequenced, that changed the game because it just exponentially increased the amount of data available for comparison. So, they are actually treated in much the same way. The morphological characters that people would use to compare, they can be looked at in the same way as nucleotides in DNA. So the AGCT, there's like millions of AGCTs in a sequence, hundreds of millions, and you can compare each of those to the next letter in the sequence for another species. So by doing that, it makes it so much more robust in a way to have this genetic code that we can compare, because ostensibly the genetic code is what makes that appearance and what makes those appearance changes. So knowing that, it made checking species identity a lot easier in some ways, and then gave rise to different problems in other ways. But it was very exciting because it meant that now there is this way to double check kind of how well our judgment of morphological characters actually lines up with this like huge database of comparison that happens with DNA.
Right. So I assume because these are obscure, there was a bigger database to begin with for all these other orders, and then this was compared afterwards?
Yes.
Okay.
Yeah. The first genome wasn't sequenced until 2012. So,
But I don't know what that compares to because I don't know when the first insect genome was sequenced.
yeah. So the first genome in general was sequenced in 1976, and it was an RNA bacteriophage.
Right. I meant first insect.
Yeah, 2000. Exactly 2000.
Okay, so 2000 was the first insect, and the first Strepsiptera was done in 2012, you said?
Yeah. So it's true that the kind of genomic revolution, as it's termed, happened quite fast. Like once we started being able to sequence whole genomes, whole DNA sequences, then the technology just kept speeding up, and it became easier and more accessible to get these codes.
So then what do you
But
do? Like, here's all the codes for the Strepsiptera, this is all the codes for beetles and flies and wasps, and who has the most likes? Like, or like similar
yeah,
DNAs? Isn't it's just that simple, or were there key ones that are more important than others?
yeah. So before we actually got to genomes, the precursor to this was just genes. Like, not just, but it was singular gene sequences or multiple that were concatenated or put together back to back. And the thing that wasn't really fully accounted for at the time was that genes have different evolutionary histories, so they can change faster or slower depending on what is being affected by the environment that they encode.
Right. It's the same reason that when you get a species into a brand new environment or quote unquote virgin environment, like arriving in
Mm.
Hawaii or something like that, or the famous Ecuadorian finches, right? Like the genetic
Yeah,
diversity explodes because they can fill all these niches and everything else. Whereas
yes, precisely.
the finch that was on the mainland may hardly change at all, but these finches on this little tiny island have this crazy changing. I assume that's kind of what you mean?
Yeah, it's similar because it's reflected in the DNA. So different genes, like a gene that encoded the beak for the finches, like maybe because they went to that new environment, and they exploded, all of like that gene changed a lot because it was advantageous for it to change a lot. So knowing that, if you compare that gene for the beak against a gene for like, I don't know, your heart function, that wouldn't change because if it changed, then you
Yeah.
would probably die.
Or you'd
um,
be behaving very differently at the very least.
yes, exactly. So if you keep that in mind and then you try to make like an evolutionary history based on the changes of that, like those two genes, they would tell a very different story.
Right.
So it was that with Strepsiptera, basically. The first couple of genes that were sequenced and used to try to infer what it was most closely related to, those told a different story than the rest of the genome eventually did later.
What? Was it
Yeah,
just by
so
pure random luck they picked a couple of genes that
it was because those were the easiest to sequence at the time.
okay, and what did those genes control?
They're 16S RNA, so they were mitochondrial genes.
Okay.
So mitochondria is the powerhouse of the cell, and they're they were genes that relate to that.
So how do we get from there? And you've already gave it away already that it's beetles they're most closely related to. So
Yeah.
what was it that they were trying to say we were more related to before we had the whole genome, and then why were we able? How did that come about? Like, what is the key that told us that it's probably beetles?
Beetles. The first, so those genes, the 16S RNA, etc., 18S too, those showed, well, the story that they told was that Strepsiptera is most closely related to flies.
Okay, so based on the data they had, they suggested the
Yes,
most reasonable explanation for it was they're closely related to flies.
yes, precisely. And then once more data were added, including the genomes, those pointed towards the story of them being most closely related to beetles.
And that's just based on the sheer number of shared genes that hadn't changed?
Yeah, or that are most similar. All of them have changed
Right,
because everything has gone through, the same amount of evolution, but the genes go through different speed of change.
right.
Okay,
So you mentioned that these twisted wings, they're similar to another fly species, right?
they're just similar to flies in general. So flies, one of their defining characters,
Yep.
all of flies, they have that hind wing that has turned into like a club. Like it looks like this.
Right. Okay.
Yeah.
Well, and for
So
those
all
that
of
are listening
Strepsiptera.
along, because there is no video at
Oh
all,
yeah, sorry,
it's
I keep
she
forgetting.
held out her arm a bit like a club that she was wielding towards me menacingly in the camera. But I was more referring, going back to the idea that is this a case where beetles also had a similar wing structure and then lost it, or is this like a new evolutionary, like two different groups evolving the same wing structure?
Yeah, thank you for asking that too. I wish I could show visuals, but I think I can guide the listeners to look up pictures perhaps. So beetles have an elytra. If people, I don't know, if anyone's listening to this and they play Minecraft or have kids who play Minecraft, then they might know what that is. But
I have no
the
idea.
understandable, even the one in Minecraft is not exactly what it is in real life, but beetles have a hard shell, most of them. That's their elytra. So that's actually their forewing, and their forewing is what has formed that case. So thinking about that, beetles and Strepsiptera then both have a modified forewing. And at first you think of things like ladybugs and you're like, well, that doesn't look at all like a club. But there are beetle families that very closely resemble Strepsiptera. So Rhipiphoridae, Ripiphoриdae, Rhipiphoridae, that is a family of wedge-shaped beetles. That's their common name. And they look a lot like Strepsipteran males. There are even some Rhipiphorids where the female stays larviform or worm-like, and therefore they have very, very similar life histories.
Wow.
So
Okay.
they're a really good case.
Okay, so the genetics solved at least the Strepsiptera problem, at least as we understand it right now. But did it also change the number of species we had identified? Like, was there a whole bunch of species that was like, actually, no, these are the same, or actually, no, these are separate species?
Yeah, that is happening right now as we speak. Like there's ongoing research and there has been very recent research that has been looking into this exact question with different groups specifically because it takes a lot of time to like analyze all of these genomes, as you can imagine. There's just a lot of data. So for the genus Stylops, which is one of the most speciose, it has the most many species in it. That group infects mining bees, or this genus Andrena. Those have been looked at by Mary Latinaro, who is a colleague, and she's really cool. She looked at Stylops and checked out cryptic species in there. So there's been kind of this overhaul looking at all of those to check out like how many species there may or may not be and what they're using as hosts and things like that. There's also a family of Strepsipterans, also the most speciose family actually, and it is made up of a family that infects wasps, all wasps, which is fun. And that is Xenidae, which is the group that we study in my current lab actually. So Daniel Benda in 2020 did this huge overhaul of that family with DNA data and found a bunch of cryptic species there as well. So there were more than expected.
So you used a term there that I wasn't expecting. I know what it means just by the nature of how you're using it. Cryptic species.
Mm-hmm. Yeah.
So what are we talking? Just species that you didn't know were there, so they're cryptic?
Yeah, that's precisely it. Because crypsis is another word for camouflage, basically. And in this context, it means they look the same. They were like hiding amongst other species, but they're actually different when we look at how much they've diverged by DNA.
Crazy. So just out of random curiosity, the parasitic wasps, are they sometimes parasitized by these as well?
Yeah, in a manner of speaking. So, it kind of depends on how you see parasitism, because I think of what I'm thinking of for a lot of them are actually like predators more so, I suppose, because their larvae are what are eating the caterpillars and things. But yeah, there are definitely like layers on layers of life history and it's very fun to think about. Even strepsipterans themselves can be infected by other things,
Sweet. A
namely,
parasite of a parasite.
yes, yes, pathogens and parasites infected themselves by pathogens. So it's a whole tangled web.
So there's insects that will parasitize these critters as well. Or are we talking
There
about
are
bacteria?
microbes.
Microbes.
Yeah, I'm talking about microbes,
Okay.
but I still think it's fascinating. I know it would have been kind of nuts if there were
Another
things like
smaller
parasitoid
insects.
wasps. Yeah, basically. Yeah, I wish. I would love that, but yeah, what I'm thinking of are things like Wolbachia. which is a gram-negative bacteria. So it's a bacteria that infects insects. It's most widely known for that, and other arthropods and nematodes, but it can be found in Strepsiptera host systems, which is something that I did during the course of my dissertation, was find this, which was really cool. There are a couple other labs who also found it around the same time. And this thing is neat because Wolbachia is known to be a reproductive parasite in insects. So it can kind of hijack the reproductive system of the insect that it's infecting. And we use it as humans, we use it to control mosquito populations.
Oh, wow.
So, yeah, if you infect a mosquito with a strain of Wolbachia and the natural population is infected with a different strain of Wolbachia, then they can't produce fertile offspring.
Crazy.
It's really cool.
Okay. I think we need to change gears because
Yeah.
we're getting really off the beaten track now, which is all good. I'm all for all kinds of different jumping off points and distractions and deviations and detours. But
Thank
we've
you.
gone through the present and the past, and now the future. So, where is our understanding for this obscure little creature going? And more importantly, is why is it important? Like, why do we need to know more about this particular insect?
Yeah. The future of Strepsiptera is just finding more Strepsiptera, honestly, because we know they're out there, and as you mentioned, there are so many species that are only known from one of the sexes, so there's definitely a lot more to be uncovered. And they just play such a complex role in their ecosystems that figuring out what they're infecting and how they're infecting them and everything is something that I think is a noble and exciting endeavor. To be fair, the why is it important is always very easy for me personally to say, but kind of hard to receive because I love them because I think they're exciting. But that doesn't necessarily get you grants.
For sure. Although you never know where science is going to lead you. And I think back to the last time I did anything bug related was actually on spiders and pheromones and how black widows find mates. And being able to figure out what pheromones exist, and you can start to control insect populations instead of gassing them in chemicals. So I would imagine that having a healthy population of these Strepsipteras is important to control different pests as well, right?
It may be. Yeah, there are actually some that infect things like plant hoppers that are found in rice fields and other agricultural contexts. And since strepsipterans, in some cases, for those plant hoppers or plant hoppers like them, can be found in relatively high levels in the population, they could be an avenue for control. You did also remind me, though, that actually, as part of this lab, one of the ways I can push for them being important and exciting is that strepsipterans, some species, it has been documented, um, or at least one species, we know that they can extend the lifespan of their host.
What? I thought they also occasionally made them infertile and things.
Yes, they do. They do, but they also extend the lifespan. So something really cool. I can't believe I left this off and didn't immediately answer with this, but something really cool about the system we study is that we're finding that the infected workers, okay, let me explain this specific system in our lab. It's between the Strepsipteran Xenos pecii and the common paper wasp, Polistes fuscatus,
The
that
yellow
is found in
jackets?
North America. No, they're different. They're kind of reddish,
Okay.
but you'll find them along the East Coast. There are other Polistes all over the US, but Polistes fuscatus is the one that we study specifically. And the cool thing about those is that in paper wasp like nests and in their social system, because they're like relatively social insect, there are workers, there are gines, and there are foundresses, and there are males. Foundresses like start a nest, but gines can they become the reproductives later in the season. So gines can overwinter and then start another nest. So they're like the foundresses. Um, Gines live longer than workers. Like workers are kind of the first wave of uh, offspring by a foundress, and they'll work to keep the nest and then care for the brood and things. But the gines are different because they have to overwinter and be able to start new nests. So we found that when a worker is infected, it lives as long as a gine. Like it is able to overwinter. And in the context of like humans, that's like a person, instead of living 70 years, living like 600 years.
Wow.
So it's 9 to 13 times longer, the lifespan.
Don't tell the anti-aging community, please. We get
Well,
enough
it's funny.
pseudoscience from them that we do
I
not
know.
need to see this.
Well, the fun thing is that University of Rochester is known for its like for aging, for investigating aging. So that's where a lot of our like fun funding is coming from, is looking into this phenomenon, because what our lab looks at are specific genes and
You should have told me
mechanisms.
that right
So
from the beginning. We would have done a whole episode on that on its own. That's
I know it
wild.
is fascinating. Yeah, it's cutting edge right now. Like we're working on multiple different projects that are approaching this from different angles, and I'm here to be like the parasite person, but they have been working on this for a number of years now in really cool ways. So they're all also um very knowledgeable about this parasite system. So I'm kind of excited to come in and also approach it from a different kind of parasite centered focus and learn about the host in the meantime too.
Sorry, I'm a little flabbergasted here. I'm like, what? That's why I love doing the podcast. You're just like all of a sudden going, wait a minute. And you start with something like, oh no, this really strange bug that I don't really understand. And yet you're like, wait, it's tied to increasing the lifespan of a particular, like, how does that affect the wasp's life cycle? Does that have any
Yeah,
ongoing effects? Like,
yeah,
okay, so that is one avenue of the future of where this is going.
it is. I'm sorry I left it out.
It's
It's so
okay.
funny
You
because
brought it right to the climax of the episode. Although people now have to listen to this point to even get it.
I know to get to it. It's funny because it's my everyday. So it's things like this that remind me that what I'm doing is actually really cool, because I'm excited about it all the time, but I forget really, you know.
But again, everything's connected to, like, every single one of these I've done that is centered around a paper. I feel like there's something there that all of a sudden I go, what? Huh?
Yeah, that
even,
makes sense.
even when I have a colleague on that's like about volcanoes, I'm still like going, wait, really?
Oh, nice.
So
There's yeah.
there's so much.
Oh,
There's just
yeah.
so much.
Science never stops giving.
There's
Oh, I was just thinking about how in Rochester and in cities really around the world, there's something called Nerd Night, and I just went to one, and the person who went was talking about geomorphology. Yeah, and she talked about sand physics. So I was just thinking about how that was a wild time.
okay. Well, I've only got one more question, and then it's starting to wrap everything up. So museums and their catalogs came up in this paper as a source, as a kind of like a sausage making of science. I don't know how I can follow up that grand finales. This is almost like the epilogue, I guess. I feel many fail to appreciate the wealth and data that many of these museums hold, both culturally and, in our case, scientifically. So just how important is that back catalog in the museums to the work that you guys do?
Oh boy, howdy. It is the lifeblood. Yeah. So museums, I don't Yeah, people don't really realize. I feel like a lot of people have been surprised when I took them, when I was still at the museum, when I would take them upstairs and show them how the sausage is made, that museums are active places of research. There are people upstairs or downstairs or behind the walls actively looking at the iceberg, the rest of the iceberg that's underwater, because the things that are displayed are just the tip. There's a fraction, a tiny fraction of the things that you see on display at museums are the collection, essentially, of the collection. And museums are kind of a library of life because taxonomy and systematics, so taxonomy describing and classifying things, and then systematics finding out how they all relate to each other, those depend on us being able to see the specimens and access the specimens for sampling, perhaps. So museums are the most important thing, I would argue, to the study of life and evolution as a whole because they provide the foundation for that research. So appreciate your local museums, but also know that there is so much below the surface that you're not seeing. And some exhibits are doing a really good job in like places like the British Museum and the AMNH especially, and the Cal Academy of Sciences, my three favorites. Those all have great exhibits about like what their scientists are actively doing, and I would encourage people to look into it because so many more people than I realized are surprised that you can work as a scientist in a museum.
Yeah, awesome. I mean, I don't get to get out to enough museums as it is, and I find closer to where I live. There's lots of cultural museums. I'm more of a science
Mm,
guy. I'd love to go to Museum of Natural History in New York and the others, and just wander around, get lost, be nerdy.
I did that every night. Well, especially for my first year there, I was just wandering the halls at 3 a.m.
Jealous.
It was awesome.
But this is also the time where you can call out one of those buddies that were on those 3 a.m. walks with you, if there was anyone, because it's time for the call outs. So this is your chance to, other work, colleagues, upcoming things, maybe a STEM group that you're, or something that you're doing in that regard of communication, whatever you got.
Oh, that's very sweet. Um, well, just because today I was thinking about them, I would love to give acknowledgement to my office mates and lab mates at the AMNH. Anna Eichert in particular she studies stoneflies and the systematics of those, and has a really cool project too with collaborating with fly fishermen, um, because they're very familiar with aquatic insects just from a different point of view. And she's going to be hopefully defending her dissertation this year too. So I'm just thinking of her because that is a really difficult journey to go through. And the rest of the Ware Lab, whom I miss dearly. Well, I was thinking about Nerd Night because I think it's cool, and things like Story Collider, so science communication, and your podcast. Like, I love doing things like this because I feel like if you're a scientist but you can't communicate what you're doing to the world, you can't get it out there, and you can't get everyone interested in learning about what you love to learn about, then it's kind of difficult to enact any change in the world. So I just want to appreciate you for doing this podcast.
Oh, thanks. Yeah, if no one understands the science, then it gets lost. Like
Mhm.
when you can't connect it back to the humans or how it affects their lives, nobody cares. And so when you can't have those conversations, it's like nobody really cares about a doctor until you get sick, right? It's the same idea.
Yeah.
You have to have those conversations because people are not going to interact with an entomologist on their day-to-day lives. But the podcast is a way where we can bring somebody into that intimate conversation about how science gets done and why it's important. So it's great. But now for something really silly. It's time for the traditional, infamous last question of the show. Tell us your favorite science joke.
Okay. Excuse me. Just so, Option number one.
Just go for it.
Okay, so this one's kind of derived from my favorite joke that I've heard my AMNH advisor, Dr. Jessica Ware, tell at a campfire. It was very funny. Here goes. There's a man and his pet millipede, basically, and every morning they kind of have a routine going where the man will walk downstairs and then the millipede will wake up and the man will like start breakfast and stuff and the millipede will go fetch the mail and then they'll have breakfast together and just hang out. But one day the mail arrives like super early. So the guy comes downstairs and he's like, "Oh my God, like I can't go outside yet." So he turns to the millipede who's just waking up and goes, "Hey, can you go fetch the mail?" And the millipede goes like, and goes to the other room. So the guy starts breakfast and he's just kind of like waiting around and the breakfast's ready and he's just like, what's happening? Like, where's the millipede? What's going on? And he's finished breakfast because it's gotten cold and he's tired of waiting. And the millipede comes back in and he's got the mail. And the guy's like, "Well, like what happened out there? Like, why did it take you so long?" And the millipede said, "Well, I had to put my shoes on."
Nice.
And then I have one that is a bit more relevant to the subject matter of this conversation.
So you told us your favorite joke, and now you're going to tell us your favorite Strepsiptera joke.
Yeah.
Okay.
Yeah. So if I were a wasp, I think I would hate to have the ability to understand like other species because then I just constantly hear the muffled conversations of the strepsipterans in my stomach. But it's no problem because I'd be able to hear them clearly in a couple days. That one was especially dumb.
They're all great. It doesn't matter. The crazier, the sillier, the less understandable, the better.
Oh, bless.
Thank you so much for coming on, and yeah, cheers.
Yeah, thank you. This was really fun.
So real quick here, because I know this episode was super long and super awesome. Loved every moment of it. But just to quickly tie back into something that happened during the episode, the first time that a species from Strepsiptera was found, they only found the females. They didn't find the males until almost a decade later for the first time. So that's really cool. Amazing. I hope you really enjoyed the conversation. I know to the heck I did. And I'll see you guys here in 2 weeks.
In a world so chaotic, so misbehaved, echoes of melodies remind us it's always colors weave stories painting the sky, swaying to rhythms as the galaxies fly by.